Hello again. In this final, or in the next video at least, I'm going to explain the processes that take place during oxidative phosphorylation. Okay? Sometimes referred to as the electron transport chain, but really the electron transport chain is only part of this process. This bit here, shown in blue, is the electron transport chain. We need to refer back to the mitochondria as a whole. So far, glycolysis has taken place in the cytoplasm. Glycolysis produces pyruvate. Pyruvate then is transported into the matrix. In the matrix is where the link reaction and Krebs cycle take place. During glycolysis, during the link reaction and during the Krebs cycle, reduced NAD and reduced FAD have been produced. So as all of those oxidation reactions take place during glycolysis, link reaction, Krebs cycle, NAD and FAD are reduced as part of those redox reactions. The oxidative phosphorylation takes place across the inner membrane. So on these cristae, and on the cristae what we find are a number of proteins that I'll explain in a bit more detail. So it happens across that membrane. So these three larger blue proteins here are, well, I sometimes refer to them as proton pumps. They're involved in actively transporting protons, H+, from the matrix and into the intermembrane space. So here, this space between the inner membrane and the outer membrane is referred to as the intermembrane space. If you are asked where oxidative phosphorylation takes place though, I would say across the inner membrane of the mitochondrion. Okay, I've spoken previously in the previous videos about how important the reduced NAD and the reduced FAD are. And this diagram is going to be similar to the one that I think you've got on page 10 in your notes, but perhaps annoyingly for you as I've got blue and red the wrong way around. I hope that's not too much of a problem. Okay, so let's start with reduced NAD. We said that when NAD, the oxidized form, is reduced, it accepts hydrogen. So it accepts electrons and protons. At the electron transport chain, the reduced NAD is oxidized back down or back to its oxidized form. And when it is oxidized, it gives up the electrons and the protons. So those protons leave the reduced NAD, and the electrons are passed from the reduced NAD onto the, or into the first, what I refer to as a proton pump. So I'm going to call that proton pump number 1. This one here is going to be proton pump number 2, and this one proton pump number 3. Now, these electrons are very high energy. Okay, they're carrying that energy from the oxidation reactions that have taken place earlier on. And the energy from those high energy electrons is provided to the proton pump when the proton pump accepts the electrons. We say the proton pump is oxidized when it accepts the electrons, and that provides the energy. that allows for a proton to be actively transported from the matrix and into the intermembrane space. And in doing so, the electron has lost some of its energy. So some of the energy was used for that active transport. Now these are just electron carriers. They pass the electron from one proton pump to the next. But that electron then passes on to the second proton pump, and it's so high energy that it allows for the movement of another proton, or the active transport of another proton across the membrane and into the intermembrane space. That electron's lost a little bit more energy, but it passes on from the second proton pump to the third proton pump in this series of oxidations. At the third proton pump, energy is provided from the electron to actively transport more protons. Now this continues until we get this really large accumulation of protons in the intermembrane space. Now this may be referred to as an electrochemical gradient. Okay, sometimes referred to as an electrochemical gradient. Electro because all of these protons have a positive charge, chemical because the protons, and a gradient because there is a higher concentration. So we get this higher concentration of protons in the intermembrane space compared to a lower concentration of protons in the matrix. So what is the purpose of all of that? Well, there is another protein at the end of this diagram, made up of two parts. This has a channel protein. Channel proteins allow for the diffusion or facilitate diffusion of ions, and this protein here, which is ATP synthase. This accumulation of protons at a high concentration compared to the low concentration in the matrix means that the protons pass down their concentration gradient through the channel, through the protein channel, and pass over the ATP synthase, and that movement of that proton provides energy to allow for the ATP synthase to take ADP and phosphorylate it to ATP. So the movement of the proton, kinetic energy, is converted into chemical potential energy, and that chemical potential energy is stored. If we remember, ATP is the something like this. That chemical potential energy that's transformed from the kinetic energy of the protons in this terminal bond, this high energy bond. So this is just a bit of an example of an energy transfer from kinetic to chemical potential. That chemical potential is stored in that high energy bond in ATP. Okay. Oops. So, you may have noticed that so far, oh, sorry, one other thing I should have mentioned. There are also inorganic phosphates, just sort of in solution in there. Okay, one other thing. This is oxidative phosphorylation. Okay. And it can only, oxidative, it can only occur in the presence of oxygen. And you may be thinking, well, we've not come across oxygen at any point during any of these stages yet. Okay, so oxygen is only present right at the very end. And it's referred to as the terminal, and this is a really important key term that you should always include when discussing oxygen's role in respiration. So oxygen acts as, pen's running out, that's what I did, the terminal electron acceptor. Okay, so use the phrase, oxygen acts as the terminal electron acceptor. Okay, but what do I mean by that? Well, let's imagine oxygen has moved into the cell from the blood, perhaps, into the cytoplasm, and from the cytoplasm into the matrix. And this oxygen is now in the matrix. Okay. Just to make this balance, I am going to add a couple more things. When reduced NAD is oxidized, it actually gives up 2 electrons. So we've got 2 electrons there, 2 electrons there, and those protons have moved from the ATP synthase down through down their concentration gradient through the protein channel. Like so. Okay. Now, what's going to happen is the protons and the electrons and the oxygen will combine together. So I'm going to call this half an oxygen molecule. Now, of course, don't get half an oxygen molecule, but it does balance my equation. So half an oxygen molecule combines with 2 protons and 2 electrons to produce water. So where we say that oxygen acts as the terminal electron acceptor, we mean it is accepting those electrons. So the oxygen is gaining the electrons, so it is oxidized with the addition of the protons as well that have moved down their proton gradient to form water, sometimes referred to as metabolic water. Without this oxygen, those electrons can't be accepted. Protons can't be accepted. And all of this would back up. And in the next video I make, I'll do a little video on anaerobic respiration to explain that in a bit more detail. Okay, so oxygen only takes part in aerobic respiration right at the very end when it accepts electrons from the 3rd proton pump. It accepts protons that have moved down their concentration gradient through the protein channel and the ATP synthase. They all combine to produce water, and the movement of those protons allows for the oxidative phosphorylation to produce ATP from ADP and inorganic phosphate. Okay, that is reduced NAD. Reduced FAD does almost exactly the same thing. The only difference is reduced FAD does not associate with the first proton pump. So reduced FAD is oxidized. It gives up its electrons to the first of those electron carriers, and those electrons do exactly the same thing. But obviously, because it's only associated with proton pump number 2, proton pump number 3, fewer protons are actually transported across. And therefore, reduced FAD is unable to produce as much ATP as the reduced NAD. In some textbooks, even in some of the older exam questions, you will see a value given for the amount of ATP that can be produced from reduced NAD and reduced FAD. It's often said, or it used to be, that one reduced NAD could produce 3 ATPs in oxidative phosphorylation. It was also said that reduced FAD, 1 would produce 2 ATPs. I don't think that's quite right, actually. I think that would only occur under very ideal conditions. And it's more like 2.5 from reduced NAD, 1.5 from reduced FAD. An exam question may give you the value, and you would be expected to calculate the amount of ATP that could be produced, but it's just a case of adding things up, really. Okay, right, that's everything on oxidative phosphorylation. I will do one more video on anaerobic respiration, when oxygen is not available to act as that terminal electron acceptor.